everybody, to the SynBioBeta webinar. My name is John Cumbers, and I'm the founder and the CEO of SynBioBeta, and I'm joining the Bay Area, California. I'm curious to find if you want to type into the chat box. This is a live webinar, and type into the chat where you're dialing in from and who you are. We'd love to know. This is a very special time for biology. Clearly, we're in the midst of a pandemic, and the global response is seeing how the synthetic biology industry and how the biotechnology industry can help to both diagnose the coronavirus or COVID-19, and also to help to come up with cures such as vaccines and antibodies and other drugs that are going to ameliorate the effects of it. So we understand that many people are working from home right now. We had this webinar planned a long time ago, and so we didn't want to postpone it. We wanted to continue to go through with it. But we realized that there's a lot of people in the industry who are working on very specific things around the response to the coronavirus. And we're going to be talking about gene editing today and how we can use the power of biology to do good in the world, both for pharmaceuticals and for many different application areas, such as plastics or materials or food and flavors, fragrances. So welcome. We're seeing somebody from Newark, New Jersey. Welcome, Gaetano. Apologies. We've got Eric from the Bay Area as well. So welcome, everybody. And we'll be doing live Q&A with Nandini shortly. So if you have questions, then you can type them into the Q&A box, and we'll answer as many as we can. And we will also be able to dial you in live if you're interested in dialing in live for asking your questions as well. So with that, I wanted to give you an overview of what's going to be coming up later on the webinar. In particular, we're going to be talking about the theme of the webinar, which is leveraging the digital genome using Inscripta's Onyx platform to accelerate forward and reverse engineering inside your laboratory. We're going to be talking about what are the current challenges of industrial and academic power users in genetic engineering. We're going to be talking about the Onyx platform and how it's a game changer. And we're going to be talking about some of the ways that academic and industrial users can employ Onyx for understanding biology at the genome scale, as well as true forward engineering of biology for many different valuable phenotypes. So without further ado, it's a pleasure to welcome Nandini Krishnamurthy. Nandini is the Senior Director of Microbial Applications at Inscripta. Welcome, Nandini.
Hi, John. It's great to be here.
Thank you.
Thanks to everyone who's dialing in.
Absolutely. And Nandini is joining us live from Des Moines, Iowa today. And a little bit of background for you on Nandini. She is the Microbial Applications Development Team Lead. She brings more than 15 years of research and development experience, leading at the interface of experimental and computational biology. And she was at various companies from Corteva Agriscience through to DuPont, through to Genentech, and has an academic pedigree. She was here at Cal at Berkeley, at Rockefeller University, and she has a PhD in plant biology from Texas A&M. So we're getting a true, somebody with true academic breadth experience and also industrial breadth experience to talk to you today. So before we the webinar, and before we start your slides, Nandini, I wanted to ask you a couple of framing questions that I had. In particular, what are some of the big current gaps that you see and the big challenges for industrial and academic power users in genome engineering?
So even though we've made large strides in genome engineering with technologies such as CRISPR, one of the key areas that I think for us to power biology is to get to genome scale discovery, right? So we are either stuck with delivering a small type of like deletion edits across the genome, or we have to restrict the depth. We can go in depth, but have to restrict the number of target loci that we can edit. So to truly power genome scale discovery, as we are seeing today, where we need to advance biology as quickly as possible, I think we need solutions that can deliver edits of different edit diversity, insertions, deletions, substitutions, anywhere in the genome. I think that is a big gap today, and we really need to solve for that.
Great, excellent. Well, I want to make sure that you're pitching your talk that you're going to give to the right audience. So if we can start the poll, we have an interactive poll that should pop up on your screens shortly. And we would like you to tell us both what's your experience of gene editing in the past, and also whether you're primarily in industry or academia or government or other. So you should see that poll pop up, and I'll share the results with everybody shortly so we can see who's online. And Nandini, going back to the framing questions that I had, early feedback on how Onyx has sped up research and development cycles for your customers. Can you give me some examples of that?
Yeah, absolutely. And I'll be sharing an example in the talk as well today, because what we see, the Onyx platform combines a suite of tools from software to benchtop instrument to novel chemistry and analytics. So what it has done for our customers, we've been working with KOLs in the field like Jim Collins and Jeff Buka, and they see this pushing their timelines from, say, months to within weeks of studying biology at the genome scale that they are not able to do today.
Got it. And can you talk a little bit about more the trade-off that researchers face in terms of depth versus breadth in genome editing?
Yeah, that's an excellent question. It's interesting. If you go and look at publications across any organisms for the genes that have been studied, E. coli, for instance, has 4,000 genes, but majority of the publications would be in like a handful of genes, right? And your RECA in E. coli or a RAD52 in yeast is probably where everybody is focusing attention on. And that's primarily because we are having to restrict our study of biology to depth rather than breadth. Or if you go to breadth, you can't then focus on a large variety at the same time. So how do you balance this where you can do genomic interrogation without having to give up on that depth is a key question to ask, especially going into interventional biology, because observational biology, yes, if you take transcriptomics, etc., people are studying that in scale, but true interventional biology where you can make the change in the genome at genome scale and study it in a parallel fashion is truly lacking today. And that's kind of the trade-off people are having to make.
Got it. Great. So we have the results from the poll, which are coming in. It says 44%, that's 20 people from academia, 24%, that's 11 people from industry, 4% from government, and 12 people, 27% from other. So thank you all for sharing in that poll. If we look at what kind of, we'll also look at your experience in gene editing shortly as well. So Nandini, what is Onyx and how is it changing the game in gene editing?
So the Onyx platform, like I said, is the first of its kind benchtop digital genome engineering platform that enables you to build gene edited libraries, cell libraries in E. coli and Saccharomyces. So usually if you have to generate the kind of diversity across the genome, typically it's a very slow process of scaling, or you work with a biofoundry where the process is done elsewhere and you get back the strains or the libraries. But what we are enabling with the Onyx platform is truly unique because Inscripta wants to democratize the CRISPR high throughput workflow, and we have a benchtop instrument and everything else that is required for a truly digital genome engineering platform that now users can just create these libraries at their benchtop and they can design the libraries that they want to study their biology of interest. So truly we take away that the component of a user having to optimize technology, so now they can focus on the phenotype that they want to study, which is the biggest excitement for biologists.
Great, excellent. And we can see who's on the line today. We have 47% are beginners, 39% are intermediate, and 17% are advanced. So I hope that helps you to know where to pitch your presentation today.
Great.
The last question before we dive into the presentation is about the software platform that underlies the Onyx platform. Can you explain a little bit about that? Because one of the biggest challenges for biologists these days is dealing with the amount of data and how to analyze it and the tools that you need. So can you explain a little bit about the informatics pipeline that Onyx has?
Absolutely. I have been on both sides, been an experimental biologist and then moved to the computational biology side. So personally, I do realize how much of a challenge doing large-scale data analysis, and even for a CRISPR practitioner, if you have to move from a single edit to moving to genome scale, a library of edits, even getting to that design, understanding annotations in the genome, comparative analytics, and optimizing guide RNA design can be a real problem because you don't know which tools to go for, what assumptions each tools are making, and what is most suited for your work, etc. So what we want to do, because we want to democratize this to both experts and beginners alike, our platform starts with software. So it's powered by robust algorithms that help you design across the genome, but also helps you analyze the data that you get back once you genotype, and you want to link your genotype to phenotype and learn through the data sets you generate to go through your next cycle of design and build and test and learn. So software is a very key component of what we are building, because we truly understand the magnitude of problems biologists can face when they are when they have to deal with this massively parallel process of getting into libraries and large designs for building these libraries and then analyzing their data.
Great. Well, Nandini, if you want to share your screen and we can bring up the slides, and then we can dive right in and show people what the Onyx looks like and how they can get started if they want to be doing gene editing in their lab. Over
Absolutely.
to you.
Are you able to see my screen okay and hear me okay?
Yeah.
Awesome.
Yeah, we got it. That's good.
So let's dive right into it. Like I was saying, we already, I'm sure most people joined by the time we had our conversation around the biggest challenge in genome engineering. And just to reiterate, most of us are right now stuck in either one of these axes, right? You can either have a small set of edit types, like say genome-wide knockout libraries in across the entire genome locations, or you're set with a small set of targets like in protein or expression engineering, where you can deliver a wide variety of edit types, let's say saturation mutagenesis or inserting multiple promoters in a small set of loci. But to get to true genome engineering and genome discovery, where you can do this in a massively parallel scale, to truly understand biology, you need solutions that deliver unprecedented scale and bring superior performance. And as I said before, we truly believe that we want to provide access to both experts and beginners. And how do you bring all of this together is what Inscripta has been working on. And here's a look at the Onyx digital genome engineering platform. Like I said before, we have developed this for E. coli and Saccharomyces cerevisiae. We are working on a mammalian platform as well. And this platform, again, allows you to generate libraries of cells, and these libraries are barcoded, and they have an ability to deliver a variety of edit types, insertions, deletions, substitution, and all at your benchtop. We have software, consumables, instrument, and genotyping kits to complete that genome engineering workflow. So let's take a quick look at how you would use the platform. I did talk about software, which is a key component. So you start with a design software, and it's called Designer. And you bring targets and the edit types you want to deliver to the portal. And these targets can be specific or they can be genome-wide. And you can say edit types such as, hey, I want to do a site saturation mutagenesis on these 5 genes, or I want to create truncations of all the proteins in E. coli. You can go from 100 to 10,000 designs per library. And that's the scale we are talking about. And once you give all those details, the software then optimizes the designs and provides you with guide RNA designs that you order. Then you order those reagents. The reagents and consumables come to your desktop. Now, what we have in those consumables are very novel chemistry and a proprietary nuclease that is a MAD7 nuclease. It's a Cas12 family, and these consumables are then loaded onto the instrument, and you push a button, and you, at the end of that process, you get a library of cells. The instrument has a lot of proprietary innovations around microfluidics and protocol optimization to give you really high quality libraries. And I'll go into a little bit more detail later on what we mean by high quality libraries. At the end of the cycle, what you get is a tube of cells, and these are edited cells and with the designs that you started with. But how do you then determine the quality of your library? We have genotyping kits that you can use. And as I said before, these are barcoded libraries. So you can use the barcode amplicon. You sequence the barcode to know what your library quality looks like. And you can also take this to your favorite mode of phenotyping. It can be selections, it can be screening. And once you identify those hits, the same kits also help you identify the genotype of the hits that you have. Because to rapidly go through discovery, you need to be able to get to that genotype to phenotype mapping as quickly as possible. Once you do the genotyping, you can bring all the sequencing data, because all the genotyping kits are designed to seamlessly work with your current sequencing workflows like NGS workflows. You bring that all the information back to our portal, where you can then do the genotype to phenotype mapping. You can compare samples and do a lot of analytics that will help you learn from the data that you've generated at scale, and then move to the next cycle. So when we look at the diversity of edit types that we've enabled on the platform, these include both edits in the coding region and non-coding region. For instance, we have inserted promoters in E. coli up to 60 base pairs and all across in front of every gene of E. coli. You can do, say, terminator substitutions at 3' end of the genes. This is an example in yeast. So this gives you an example of how you can build libraries that regulate gene expression in different ways. And in the coding region, there's an example of how do you create a knockout to say, let's say you want to study loss of function. There's way of truncating proteins by inserting a triple stop codon, or your typical saturation mutagenesis library that you can study across a given protein. And these are just demonstrated edit types, and I'm presenting this to you just to trigger your imagination of what you would do given this kind of power at your benchtop.
And Nandini,
yeah,
I want to interrupt a second. Could you jump back to the to the slide before? So I know we have a lot of beginners in gene editing on the line. So just looking at this designed generate, test, learn cycle that we're going through. So at the moment, if people are doing gene editing in their lab, in their company, or in their academic lab, excuse me, it's quite a slow process. They're doing mostly single gene edits at a time. Maybe they're designing it on some piece of software that they're using. That's not maybe customized for gene editing applications, and the throughput is very, very slow, and the design of the experiment is very laborious. And what you're offering here with this piece of equipment is a desktop device that sits on your bench, and a integration of software and hardware and wetware, and it's all inside of this box. So you design your let's say you're wanting to make an amino acid, or you're wanting to improve the yield of an amino acid, for example. So you can take that and you can design your pathway, and you can design the set of edits that you want on the software. Then the reagents will come into come into your lab. You put them into this machine, and then a given number of hours later, out pops this cell library of cells that you can then use in your downstream screening applications to find which of those might produce the amino acid or increase yields of the amino acid. Is that correct?
Absolutely. I think you summed it up really beautifully, John. Thank you so much.
Sure.
There's One area that I want to highlight at this point, I want to highlight that these are single edits we are delivering. So these are massively parallel single edits that we are delivering in the cells to enable diversity generation, and we can go up to 60 base pairs in length, and we are working to improve both fronts. Like, we are working on a combinatorial platform, and we are working to increase the edit lengths that can be delivered with efficiency as well.
Excellent.
So I just wanted to make sure our audience understood the concept there.
Great. And I just want to remind people this is live, and if you have questions, you can type them into the Q&A box, and I'll either ask them for you, or we'll ask you if you want to ask them to Nandini yourself. So sorry to interrupt you, Nandini, but if you want to continue.
Hey, no worries. This is great. I'll have to work through all these animations again. So let me just take this off. And we talked about scale, John, just now, as to, you spoke about how most processes are low throughput. People are definitely trying to scale up the CRISPR process. And a predominant way we are trying to do this these days is through a microtiter plate-based process, right? So typically, if you wanted to create a 10,000 member library, you would have the problem of first going to the software to figure out which software, how do you design for 10,000 targets? But then comes the problem of, okay, I've designed the guide RNA, and now I also have a donor that I want to use as a template for HDR. How do I co-localize all of this? So for a large-scale library in a microtiter plate situation, it's a painful process to co-localize each of these, and requires investment in infrastructure and automation if you want to truly scale this process to, say, 10,000 member library. And you will end up probably spending time with PCR assays to track each of these edits that and how they went, how they were delivered. Now, one way of increasing scale here is to move from a well-based or addressed build to a pooled format where you use cells as the partition. So immediately you break this the barrier of scale and go from, say, 100 build to 100,000 build in no time. But the challenge then is, how do you co-localize your guide RNA and donor in a pooled setting? So our co-founder, Andrew Garst, actually came up with a very elegant solution where he covalently linked the guide RNA and the donor, and that solves that co-localization problem. But what he went ahead and did was also add a barcode to this construct. So now what you have is the ability to create large libraries of scale where you have the guide RNA and donor co-localized and barcodes available to track what's in the library. So you can track your edits through this. So this talks about scale, but then one thing to keep in mind is when you go into this pooled kind of build, it brings up some unique challenges of performance. And let me just walk you through what they are. So what you see here is typically, let's say in a scenario for yeast, you have a lot of labs that work on a single edit paradigm, and you often see reports on single edit efficiencies. So what do I mean by edit efficiency? Edit efficiency can be defined as a fraction of cells in your library that contain an edit. So which means, what is the usable fraction for you that you can actually use and will give you returns in your phenotyping paradigm? But one of the things that we see in literature is often people report, say, an edit efficiency of 90% for a given design. And when they move to a pooled format where you have a library of designs, they extrapolate that same edit efficiency to that library. But what we have seen repeatedly is when you move from a single edit to a library format, edit efficiencies fall from 90% to as low as they fall to like single digit percentages, like 5% or 2%. And so that is a huge problem. Why does this happen? Two reasons, primarily. One, not all guides are created equal in their efficiency to edit. And Once you get to a library format, the nonlinearity of biology kicks in, and the cell populations can then get burdened with what we call the SKP problem, where there are a lot of non-editors that take over the population. So then the actual usable fraction of cells is very, very low. So Inscripta is actually has worked to optimize both those problems. The first, the design efficiency is solved by really good algorithms to improve designs across the genome. And the second is the instrument, which has proprietary microfluidics and protocol optimization that manages this nonlinearity of biology to give you really high-performing libraries. So if we look at the design software, as I said, there is really robust algorithms that we are using. We've done a lot of optimization around guide design, and we have a lot of empirical data that we have now used for modeling to predict guide efficiency. And you can see a lot of features that there's more than 1,000 features we've evaluated, and we are continuously improving this. But in addition to design and improving quality of library, how do you know you have a good quality library? So genotyping and quality control is very, very important. So we have very stringent metrics. I spoke about genotyping assays that we have at the barcode amplicon, or even we also have assays that measure edit at the genome level, genomic level. I want to introduce one new concept here called the complete intended edit. So in a library, when I say you have what is the fraction of cells that are edited, what we count is a very, very stringent fraction. That is what we call as complete and intended edit. Let's take an example here. You have a wild type sequence. Let's say this is TAC, is a codon that codes for a tyrosine, and you want to convert it in your design to ACT, which is a codon for threonine. We also add a few edits, auxiliary edits, to prevent recutting by the MAD7 nuclease. Now, we only count those edits that match your design, even though other entities exist in the population. Some of them could be complete but unintended, like in this case, where you have ACA, which also codes for threonine, but is not the intended change. And some of them could be completely partial, and there could be others that didn't edit at all. So it's important to be able to evaluate a quality of a library where you have stringent metrics. And we also provide other metrics that helps you answer two very, very critical questions for anyone working with libraries. One, what is the fraction of cells that have edits in my population? In this case, complete intended edits, which we call edit efficiency. And the second is, what fraction of designs that I designed, that I input into the system, are represented in the library? Let's say I started with 1,000 designs. Do I see all of them in the library, or do I see only a subset of them? So with edit efficiency, we've been able to achieve, remember I told you that we can actually drop to single-digit edit efficiencies when we go from single design to library of design. We have been able to get it to an average of 30%, both in E. coli and yeast. And this is not accounting for fitness effects of targets, right? If you're studying essential genes, of course, you will have some fitness effects. The range is anywhere from 15 to 75% in the edit efficiency. And when it comes to representing designs in the population, we consistently see very, very high representation of design in the population. We observe designs up to 96 to 99% consistently. What you see in the graph here is a quick snapshot of edit efficiency. And this is an example in E. coli libraries, and I selected a set of libraries to showcase the differences in edit number of designs per library. The first few are close to 100, then you see 1,000, and finally you see about 10,000 designs in the library. And the pink bars here are the complete intended edits, and the gray is the wild type or that partial component or an incomplete edits that we spoke about earlier. The y-axis shows you the edit efficiency, and you can see that it varies depending on the targets you choose, the guides that you choose, etc. And but overall, you can see really good edit efficiencies in these libraries, thanks to both the software and the innovations around the instruments. So that's kind of a quick overview of the technology. And John, do we want to take questions now, or I can move to applications and talk through how people can use the platform?
Yeah, if people have questions, then please type them into the Q&A box on the webinar, and you can also hit the raise hand button if you want to ask your question live. Nandini, I have a couple of questions. One is just looking at the big picture here. What's the current scale that people can do gene editing, and then what does the Onyx allow? And then how many cells would you get out of a single run on the Onyx, or what's the range that that the piece of equipment allows?
Right. So the Onyx currently allows you to create libraries anywhere from 100 designs to 10,000 designs in E. coli, and 100 designs to 6,000 designs in Saccharomyces cerevisiae. So this really helps to scan the entire spectrum of needs that customers might have in terms of the biological question they want to answer. And the number of cells that come out, we have anywhere from 10 power 6 to 10 power 7 cells that are generated at the end of the cycle on the instrument. That gives users a quite large set of cells to go forward with for either screening or selection protocols.
Great, thank you. And we have a question from Jose Utrilla, who says, what is the limit of the length of the insertions?
Yeah, at this point, we have 60 base pairs as the edit length, and that will be the first offering, and we are working towards improving this over time.
Great, excellent. If you want to continue.
Sure. So what can you really do on this platform? The application space, as you can imagine, is wide open, because what the platform does is provide you with an opportunity to create the kind of diversity that was not possible today. Whether it's, you can study strain engineering, metabolic engineering from an industrial perspective, or study fundamental biological phenomena, signaling pathways, genetic interactions. You can study any of these aspects using the Onyx platform. So let's look at a few examples of how you would do this. So I'll start with framing more around what kind of phenotyping workflows customers are likely to have, right? I'll start with the pooled selection workflow. And here's where the having barcoded libraries is really, really an advantage. Let's say you're studying, for instance, antibiotic resistance, and you create a library on the Onyx platform, which has a wide set of diversity. And you can then go into pooled cultivations here. You can treat these cells with a variety of different antibiotics, study a time course. And at the end of it all, you can then use the barcode, sequencing of the barcode, to quickly identify which of these edits respond to the stress that the treatment that you have provided in your experiment. And the software then allows you very quick genotype to phenotype mapping. So in a very rapid time frame, within weeks, you now have the ability to identify a wide variety of genotypes in parallel in a pooled selection. So just to illustrate the power of this, I'll show you a snapshot of an experiment we did. This is to study genome scale target discovery for osmotic stress. We created 7 different libraries, which had a very different set of edits, that anywhere from loss of function to gain of function. We had a range of promoters that we had inserted to study how that would respond to the stress we were giving. And we also had knockouts, typically truncations, protein truncations. And this was at genome scale on all different genes of E. coli. We put it on we treated them with 4 different concentrations of salt and 3 different replicates. So what we then did was did a quick barcode sequencing to see what designs enriched and what designs depleted. And across the different regulatory paradigm that we have, and across different salt treatments for all 4,000 genes in E. coli. So this is truly a powerful way of studying genomes at scale and to discover new biology, and to go truly from being just observational in biology to becoming interventional and potentially studying, getting to causal genes really, really quickly for the phenomenon that you're studying. The other paradigm in phenotyping typically is screening, where many of you probably are looking at isolating colonies and looking for typical, say, a metabolite screening for every colony to identify hits. The Inscripta platform also plays seamlessly into that kind of a workflow. So I'm showing you here an example typically of a workflow we've used where we built the library and you get a pool of cells at the end of that process. And you can use typical infrastructure like colony pickers, liquid handlers, and then link it to what we've done is an Agilent rapid fire mass spec to study critical metabolites that might be changing in the library that we've created. So let me go through a quick example of a genome scale metabolic engineering story. We took the native genome of E. coli and created edits across the genome to ask, can we study overproduction of lysine? And as you know, lysine is a very, very well-studied pathway. So the first question was, what do you target for these edits? And we started, we said, okay, let's use a rational approach first, where we take the pathway of lysine biosynthesis and some ancillary peripheral pathways that feed into it. We took all those enzymes and did a saturation mutagenesis. But we also said, why don't we go to the other extreme and take a completely unbiased approach and go through a genome-wide target set. So we created about 24 different libraries where the rational approach had 20 targets. Unbiased approach is, of course, the entire genome of E. coli, which is about more than 4,000 genes. And various different library strategies such as saturation mutagenesis, genome-wide promoter ladders, 5 different promoters so that you can tune expression, and the truncations as well. And the idea here is you create a large swath of diversity, and then you sample in a shallow way quickly from all this diversity libraries that you've generated. So you end up getting a unique set of edits that you can discover as hits in your process. And in this case, as I said, we used the rapid fire mass spec to look for lysine titers in each of these colonies that we had isolated. The graph here shows you on the x-axis the different libraries that we created. The center panel is typically the very targeted approach that we had, a rational approach of all the pathways and site saturation mutagenesis, and it's bookended by the unbiased approach where you have the truncations and the different promoters that we had inserted. And the y-axis is the log fold change with respect to the wild type. This is a primary test, and you can see we already have hits. So we took the typical top hits from all of these and retested them on both minimal media and rich media. And what was interesting was we recapitulated a lot of the hits from the lysine pathway that's already been reported in literature. So that was a great step for us. But what was truly interesting is that we identified new targets which were not in the lysine pathway. And I think this is truly possible because of the kind of diversity, edit diversity you see, and the targets we were able to go after. Interestingly, a lot of the targets were from the biome of E. coli, which is the genome that is not very well annotated. For any forward engineer, though, you don't stop with diversity generation, right? You want to be able to combine this. While we are working on a combinatorial platform, we ask the question, can you actually stack these mutations together? So here's an example where in the first round I just spoke to you about, I'm highlighting 2 different variants that gave us a huge fold improvement over wild type. One was a protein mutation in DAP-A, and the other was an insertion of a promoter in front of DAP-A. So we took this very well-known amino acid substitution. So we took this strain and asked the question, if we rebuilt the library on this, do you see an improvement in phenotype? And you already see that addition of the DAP-A promoter swap gave you a 10,000-fold increase. And when we took that strain and moved to the next step, we got a 14,000-fold improvement with a new edit added onto this set. It's not surprising that you get a quick jump in phenotype, especially when compared to wild type in this case. But what is important to remember is we did this in a matter of 5 weeks. Of course, the vision is to go from a truly combinatorial fashion, where you go from round 1 to round 3 that completely combines all these edits to get to a phenotype that is much higher than where you started from.
Nandini, if I can just pause you a second and again help to translate for those beginners who are dialing in.
Mm-hmm.
So what we're seeing here, and you're using the example that I gave earlier, which is the lysine production pathway. So lysine is an industrially relevant amino acid. It's used in animal feed. It's an essential amino acid. It's used in food products, and it's produced via fermentation. So sugar into a yeast cell produces lysine, and it's been studied for decades, the different pathways that produce lysine in yeast. Is that right?
Sorry, John, I missed that last question. The audio dropped.
I was just confirming that lysine is been studied for decades, the lysine production pathway for the conversion of sugar, glucose into lysine. And then what you did was to create a library. So you knew the lysine pathway, you knew the genes, the number of enzymes that were connected together to produce lysine, and you then created a library of mutations along that pathway. Then you screened for the phenotype, for the production of lysine, and you found that you could recreate all of the previous studies that people had found in the lysine production pathway from the scientific literature that they'd done by knocking out one gene at a time or looking at overexpressing particular enzymes along the pathway. But then in addition to all the ones that you found that were in the literature, you found a whole bunch of new possibilities for increase in the production of lysine that nobody had ever come across before.
Yes, and that was possible only because we took an unbiased approach, right? Typically, what happens is you're either you're restricted. This is that depth versus breadth question we were discussing earlier. If your solutions don't allow you to scale, you are always looking in known pathways and prior hypothesis. It's like, where is the light that I can look for the key? You cannot expand this beyond that. So what we wanted to showcase was, even in a really well-studied pathway, when you expand it to a genome-wide, completely unbiased approach, you still have targets that give you hits, which now when you combine can further improve your phenotype. So I think that's a very key point for the audience to understand.
Got it. And can you just explain again, in terms of the quantity, how many edits were made in the lysine pathway? What kind of edits were they? And then how many phenotypes were produced again? Or maybe you're getting to that.
Yeah, so I just want to touch on this here. So we took both, like I said, rational and the unbiased approach with a multitude of targets. It was 24 libraries, and we made about 200,000 designs. And this is, the 200,000 is not in a single library. So I do want to say these are different libraries, 24 different libraries. And you had a wide variety of mutations here. So what we wanted to showcase in the study is using a diversity of edits, where you are looking at protein function with site saturation mutagenesis, you're looking at gene expression regulation with the genome-wide promoter ladder, and you're looking at loss of function, where you find other hits that comes through. So you need a combination of biological knobs, let's say, to tune your system where you can get to the best possible phenotypic landscape.
Got it. Great. Thank you. Thank you for clarifying that.
Awesome. Thanks, John. Um, so I want to switch to a second study, which is very different from the first, and now in yeast. And this is in a pooled selection format of phenotyping. Several of you, when you're engineering or studying a strain, can find that over time, the strain starts accumulating mutations, and it's very hard to debug which of these and these accumulated mutations can give you weird phenotypes, right? Poor growth, etc. So one project that we are working on with Dr. Buka at the NYU is around how do you optimize, yeah.
Sorry to interrupt you. I just want to remind folks listening that this is a live and interactive webinar. So if you have questions of anything that Nandini talked about in terms of the lysine production pathway or the hardware or this next project with Jeff Buka, then type them into the Q&A box and or raise your hand and we'll get to your questions. We'll also have a Q&A period at the end. So sorry to interrupt you, Nandini. Please continue.
Hey, no worries. Yeah, it'll be awesome to get questions because that's how we also learn of what our needs are for the users. So coming back to the SE 2.0 project that Jeff is the director, it's an international project where there are multiple groups involved, and the goal is to complete the synthesis of a custom designed genome. And the model system they are using is Saccharomyces, and each group has taken on the incredible task of synthesizing every chromosome of yeast. So they're redesigning and re-engineering one chromosome at a time to finally create a completely synthetic organism. So the challenge here is while engineering, they are also trying to introduce a lot of different markers or PCR tags, etc., using the typical scramble technology. So what Jeff's team saw, they're working on the synthetic 6 chromosomes, so Syn6 strain. And typically over time, it starts accumulating both maybe because of unintentional change, or sometimes even the changes they're making ends up creating a slow growth phenotype. Now to debug this, they had to go one mutation at a time, identifying what it could be, correcting it, and checking, okay, does it improve the phenotype? And months and months of work, they identified 4 phenotypes four changes that causes this slow growth. So the challenge they pose to us is, can you use your technology to rapidly reverse this, identify which ones could be causing the slow growth phenotype and help reverse that? So they gave us a set of changes to make where this table shows you the kind of changes they have engineered into the Syn6 chromosome. And the question they asked is, can you go back into the Syn6 strain and revert all of this to the wild type? And we did not know which ones of these will give you a better growth phenotype. So we went in blind. There were about 400 odd targets, and we made redundant design. So that is something Inscripta platform allows. You can make more than one design to the same target. And so we made about 900 or 1,000, about 1,000 member library in yeast. And then we ran a typical growth selection. And what you see here is the barcode amplicon data. So we sequenced the barcode, and this is a typical volcano plot where you have the log-fold change on the x-axis and the adjusted p-value on the y-axis. And you see this particular quadrant that shows you barcode counts where compared to the original time 0 at 24 hours, you see a huge enrichment. What this means is that both biologically and statistically, you see these edits in the population overrepresented because they were potentially having better growth phenotype. And interestingly, what we were able to do is recapitulate all those mutations that they had originally identified over time, one by one, in one experiment within weeks. We were able to identify that. We also have other candidates here that are potentially also causing, which would improve their growth phenotype, that they are currently revalidating in the lab. So this is a good example of how you might use the Inscripta technology in a pooled format and maybe for debugging your strain. So I'd leave you with some of the specs here. We've been talking through this. So here's a summary. This is the platform developed for E. coli and Saccharomyces, and 1 edit per cell with insertions, deletions, and substitutions. The scale is 100 to 10,000 designs per library for E. coli, 100 to 6,000 designs for Saccharomyces. So in a span of a few weeks, now you can actually design and generate libraries in scale at your benchtop to create the kind of diversity that you want anywhere in the genome for any of the different edit types. So thank you so much for your attention, and looking forward to your questions.
Excellent. Well, thank you, Nandini. We appreciate the depth that you've gone into the Onyx genome editing platform, and we do have a few questions here. S. Chang says, for the lysine pathway, slide 18, how were the 24 libraries screened for lysine production in the rapid fire? Was each cell isolated and incubated? How long did it take?
So yes, so this is the kind of workflow I think you're referring to, Chang's. So each of these, in this case, we do have the infrastructure to isolate cells, and we have a protocol where we grow them over time in the required media, and then finally we do the rapid fire itself is a very, very fast process. It's 7 seconds a sample. So in a week, we can process hundreds of plates here.
Great, excellent. And then Najin has a question. Were those mutations causing the 14,000-fold improvement in the lysine production somehow related to lysine biosynthesis genes?
So some of them were, but we do have where we have the 14,000-fold. Like 2 of these were definitely related to lysine production. DAPE is a very well-known target. And of course, we did select the strain containing these edits. The GlenD is not a direct candidate in the lysine biosynthesis. And at this point, we don't know how it relates to lysine biosynthesis, because in this particular paradigm, what we are looking for is how quickly can you get to the next phenotype and not study individual gene function. But you can use the platform to do that as well.
Great. We're getting some really interesting questions coming in, so I hope we'll have time to get to them all. And just a reminder, this is live, and if you want to raise your hand in the webinar, we can call on you and you can turn on your microphone and ask Nandini a question directly. And if you're feeling brave and you're not in your work from home pajamas, then you can also turn on your video, and we'll ask you before we put you on a video. But otherwise, just raise your hand and we'll come to you on audio. We have a question from Merv Nida, who says, does Onyx perform rational design or random design, or both?
It's both, because I wouldn't say random design. I think the target space is both rational and random, and you can design any kind of edit type, whether you want it to be rational or random. The choice is yours, and you can combine both too, like we did in these experiments.
Great. And Mijay has a question. For the lysine example, was any regression analysis or machine learning approaches used for recommending combinatorial edits for the next editing round? Good question.
We are working on that. At this point, we do have the capability to do use machine learning to get to combinatorial approaches, which is very key, especially when you do this in large scale. So we are working towards offering that as the next round in our software roadmap.
Okay, excellent. Jose Utrilla has another question. How easy or difficult is it to move to a different host, let's say Bacillus subtilis or Pseudomonas, or a different kind of yeast?
Yeah, I think this is a question we get asked very often. So what we're doing at this point is trying to understand what are the different highly used hosts in the community that we can start working on. The typical areas to optimize would start from design, understanding the genome, then understanding the editing efficiencies of the host, the growth properties, transformation, etc. So it's hard to predict a timeline to get to that, but definitely this is something we are looking into.
Great, excellent. And we just have a few more questions. So if you do have questions, please type them into the box and we'll try to get to as many as we can. And please raise your hand if you want to speak to Nandini live. Aki Ueza has a very interesting question for the future product roadmap. Aki is asking, well, can mammalian cells be used in the Onyx platform?
Absolutely. So what we are working on is we do have a mammalian team that is developing a massively parallel editing platform, just like the Onyx for microbes for mammalian cells. So that is definitely a future offering we're looking at.
Excellent, great. So Nandini, we're coming to the end. If you want to unshare your screen again, I just have a few other questions for you. If you can talk a little bit about how the genotyping assay works for Onyx. For example, I know Inscripta has anticipated the need to genotype the winning edits that users recover from a screening campaign. And these screens could be done with several existing tools, including mass spec or a fluorescent plate reader, or microscopy, or genome interrogation. But Inscripta offers a portfolio of assays designed for Illumina sequencing. And Inscripta CEO Kevin Ness has said recently that his assays could theoretically be designed to work with other platforms too. So could you just delve in a little bit deeper into the genotyping assays?
Right. So genotyping is a very critical aspect for customers. Your phenotyping landscape is infinite actually, but in all of these cases, one of the key handle for us is the barcode. And what I want to emphasize is we purposefully decided not to add the barcode to the genome, so it is external to the genome on a plasmid. So it gives you a quick read of what kind of edits is driving your phenotype using that barcode amplicon. It's a very easy assay. It's a typical NGS assay where you have a very small barcode, about 10 to 12 nucleotides, and you sequence it, and that fits. And the idea is that it'll fit with everybody's workflow, and NGS being so popular that everybody uses it these days. Now, if you're running a screening assay and you are only targeting, let's say, a single gene but going in depth, if you have genomic amplicon assays where you do your PCR amplicon and sequence it, those can be used with our platform as well. So if you have existing assays, those can be incorporated into your workflows as well. So we give that level of flexibility.
Great. And Abhinay Badri has another question. Could you explain the design features in the software? Is incorporating metabolic network analysis something that you have in the future product pipeline?
Absolutely. So this is something, so at this point in time, we do not have anything that enables customers to do target discovery. So we start with customers coming in with target discovery, and we focused on design performance. But the next offering in the roadmap that we are working on is how do you improve target discovery, and what methods can you use for both metabolic engineering or other biological phenomenon that you want to study.
Great. And Merv Nida asked a question, does Inscripta offer sponsorship for iGEM teams? And I guess that's a broader question about your engagement with academic labs.
Yeah, so we have a lot of different plans where we are looking at a very broad engagement and building a community around the Inscripta platform. So it's hard for me to go into details right now. So maybe if the user can contact us, we can provide details around the plans that we are thinking of.
Great. And what's the best way for people to get in touch with you, Nandini?
So we do have a website, email address that is info@inscripta.com that they can write to.
And the last question that we have from Jose Utrilla is the platform available as a service?
No, the platform at this point, I guess the question is, do we offer services to build libraries like a biofoundry would? No, our model is very different. We do want to get the technology in the hands of our users, and that's why that greater access in the beginning of the seminar was a very important piece for us. We want customers to be able to control what they build and how they build it. So at this point, we do not have a service model.
Got it. Well, thank you all for joining us today. I hope you've enjoyed the interactive Q&A and enjoyed the overview of gene editing and what the Onyx platform can do. Thank you, Nandini, for participating. That was a great overview that you gave us. And this recording will be available at synbiobeta.com/Onyx. So we hope that you'll join us there. And we hope that you'll join us at the conference coming up in September. And we will all be able to get hands-on experience with some workshops with the Onyx and a number of other activities that Inscripta is going to be sponsoring at the conference. So thank you, Nandini, and thank you everybody for joining us today. We hope that you enjoy the rest of the day and the rest of your week. Stay healthy.
Thank you, John. And yeah, thanks
Thank you,
everyone.
Nandini.
Stay safe and stay healthy.
Cheers. Bye-bye now.
Bye.